All right, so as we wrap up section 5.3 of the Pierce text, we're going to talk a little bit about genomic imprinting because this is vastly becoming a very intense area of research, especially in agriculture. So, looking at the different ways that the genomics can actually be influenced by the environment that the individual is actually gestating in. So we see that this a lot taking place in animal research. It's called fetal programming to figure out exactly what is going on relative to each individual genotype. All right, so genomic imprinting is defined as a differential expression of a gene that is dependent on whether it is inherited from mom or dad. So, an example of this, as described in the book, is the insulin like growth factor 2 gene, or IGF2. And this gene is responsible for directing nutrients through the placenta to the baby during gestation. So you can see that if there was a mutation in this gene, it would actually result in like premature babies or even miscarriages. So this gene is found on chromosome 11, and it's an autosomal gene, but it is actually determined based on which expression or which allele is being expressed, whether it was from mom or from dad. So, relative to IGF2, the paternal copy is always going to be dominant and the maternal copy is silent. So, the paternal genotype of IGF2 can determine the growth in gestation of these babies. So to demonstrate this, has a really nice figure in your book. This is figure 5.18. And in the first segment here, we've got a paternal allele shown in red and a maternal allele shown in blue. And each of those alleles have different copies of the IGF2 allele. So as those copies come together during fertilization, the paternal allele or the red allele is going to be the active allele, and its protein that is generated through transcription and translation is actually going to stimulate the fetal growth in gestation. So the maternal allele ends up being silent, and so the absence of its protein product does not further stimulate fetal growth. And so the size of the fetus is determined by the combined effect of both alleles. So just keep in mind that this is happening on chromosome number 11. So the IGF2 gene is actually up near the telomere. And so you could see, as we talked through chapter 8 and chapter 10, if chromosomes are not inherited correctly, that could be one gene that could be heavily mutated. And so that could be one of the reasons that we're seeing some incidents of miscarriage among certain individuals. All right, so typical Mendelian law states that if we have two loci that are determining two different traits. So, for example, the one that we've been using in chapter 3 and chapter 4 is the brown phenotype and the yellow phenotype in pea plants. And so, if we cross the dominant form with the recessive form, then we know that we're going to get a 9:3:1 phenotypic ratio. So, that is typical Mendelian inheritance with dominant and recessive inheritance patterns. So this type of pattern is shown through independent assortment, which happens during meiosis. And independent assortment says that alleles are going to act independently with regard to phenotypic expression. So in other words, the shape of the seed absolutely has no effect on the color of the seed and vice versa. With genomic imprinting, that law kind of gets thrown out the window. So autosomal genes, both in males and females, make an equal contribution in terms of total genetic material, and it is thought that they should have equal effects upon the phenotype. But in reality, that's not always true. So some genes are only going to be expressed if they're transmitted by the male pet parent or the female parent. And so that's the example that we see with the IGF2 gene. Some other syndromes that are results of genomic imprinting include Prader-Willi syndrome. So these individuals are typically going to be male, and they're going to have very small hands and feet, be very short, and have severe mental retardation. They also tend to be heavier set or obese. And what ends up happening with these individuals is that they have a small piece of chromosome 5 that is actually missing. So it's a deletion, and that deletion is always inherited from the father. A second syndrome is Angelman syndrome, and this one we see usually in girls, but it's seen in boys too. And Angelman syndrome is where the individual has frequent laughter that can't be controlled. So they have infrequent and uncontrolled muscle movement. They tend to have a very large mouth, and they tend to seizure quite often. And this is also isolated to chromosome 5, and specifically a deletion on chromosome 5. And these individuals end up with this syndrome, and they will inherit it from the mother. So, two different syndromes, both on chromosome 5, and very different phenotypes depending on whether they inherited the gene from mom or from dad. Some other syndromes that are really starting to trace back to being or the result of genomic imprinting include bipolar disease, carcinogenesis, and a slew of others. So there's about 25 total syndromes that have been isolated to genomic imprinting. All right, so as we finish up section 5.3, this table really gives you a good breakdown of the different genetic phenomenon that we've been talking about, whether it was sex-linked or genomic imprinting, and it gives you a description of how the phenotype is determined. So this is a good place to start studying.